What's up, Ninja Nerds? In this video today, we're going to be talking about the lysosomes. We're going to talk about their structure, and we'll also talk about their function. But before we get started, if this video benefits you, it helps you, it makes sense, please support us. And one of the best ways that you guys can do that to help us to continue to make these awesome videos for you guys' enjoyment is by hitting that like button, commenting down in the comment section, and most importantly, subscribe. Also, great notes, illustrations that our team has come up with, you guys can check out on our Ninja Nerd website. I'll have a link down in the description box where you guys can go check that out, all right? But without further ado, let's talk about the structure and function of lysosomes. So nothing crazy here. When we talk about lysosomes, one of the things I want to do is recap a little bit of things that we talked about with the rough ER, things that we talked about with the Golgi, things that we talked about with the cell membrane. So it's going to be somewhat of a recap, but it's okay. That's good. That's what helps you guys repetition to understand these things. So when we talk about lysosomes, what I want to know is their structure, but how are they made? All right, these are really interesting. So when we actually talk about lysosomes, it all starts at the nucleus, believe it or not. So in the nucleus, you guys know that we have chromatin, and one of the components of chromatin is DNA. If we were to take DNA and read it and make a single-stranded structure called an mRNA, that would be called transcription. So from this DNA molecule, we're going to make something called mRNA. And this is via the process of transcription of the DNA. Then the mRNA will then go and bind with a ribosome. And what we know is that ribosomes can exist in various forms. They can exist in the cytosolic form, in other words, just a free form which floats in the cytosol. Usually that one is to make proteins and enzymes that are functioning in the cytosol or that go to the mitochondria. But the other ones are membrane bound to the rough endoplasmic reticulum. These are making proteins that are designed to go to three particular locations: the cell membrane, to be excreted, or to become lysosomal proteins. You're getting a point here, right? So once we take this mRNA, we bind it with the rough ER ribosomes, the rough ER ribosomes. Once they fuse here, they start to synthesize a particular protein. So there'll be a protein synthesis that'll occur here in the actual ribosomes. Now, once that occurs, here's my protein that got synthesized in the ribosome. Then what it's going to do is it's going to start making its way through the rough ER, where it'll have some further modifications. Do you guys remember what we said occurs here? The end-linked glycosylation will add a sugar residue to asparagine, one of the amino acids on this protein. Then what we'll do is, once we've modified this protein, so let's say here we modified a little bit. Here's the core protein, and then all we did to this core protein is we added a little sugar molecule there. Then what we do is we're going to package it into a vesicle out of the rough ER and say, hey, you now need to go to the Golgi, because the Golgi's got to modify you a little bit more to make you very, very specialized and determine where you're going to go. So then we make a little vesicle, and then how do we get that vesicle to go to the Golgi? Do you guys remember what was that pink protein that we put on its surface that helps it to guide it towards going to the Golgi? What was that protein called? This was the COP2 protein. All right, just look, checking your understanding. Once this vesicle fuses with the Golgi membrane, it then releases the protein contents here, this modified protein at this point, into the Golgi apparatus. And then in the Golgi apparatus, it'll be further modified. And when it's modified, then what will happen is it'll then get packaged into vesicles in the Golgi that'll be sent to three particular locations. Out of these, let's say here is the three particular locations. Here's the protein. The proteins are all synthesized here. Some of the proteins can get packaged into these vesicles here, right? And then from this can go to three particular locations. One of them is they could be excreted from the cell. So these could be an excretory protein. We know this already, right? So this is pretty obvious that this thing could be excreted. So here's my excretory protein. The other aspect of this is that protein that's been modified in particular ways can also be incorporated into the cell membrane. So here's my protein that's now been incorporated into the cell membrane. And again, all of these have these particular sugar residues on them. The last thing is they could become lysosomes. Here's where we have to add another quick fact in. We talked about in the Golgi that there's three modification mechanisms, right? In the rough ER, there was one. It was end-linked glycosylation. In the Golgi, there was trimming of the end-linked glycosylation. There's O-linked glycosylation, so we add a sugar residue onto the oxygen of serine and threonine. Or there was phosphorylation. This is the key one that I really want you to remember. There's a sugar that's present on the sugar residue. There's a molecule called mannose. When we phosphorylate mannose So we call this phosphorylation. I'm going to write this down because this is a crucial step here. So we actually do phosphorylation of what's called mannose, which is basically one of the sugars that are present on that long sugar residue that was attached. We put a phosphate on it. Once you do that, you then say these proteins, these glycoproteins, are now destined to go and become a lysosome. That is the thing that I want you to remember. Okay, so when we talk about lysosomes, lysosomes are really just these spherical vesicles that have a membrane that was formed, an actual phospholipid bilayer that was formed by the Golgi, and they contain glycoproteins that were synthesized from DNA going to RNA, going in to make proteins, modified in the rough ER, and then modified in the Golgi. In what particular way? What was that big modification thing that I told you? This is super crucial here, is phosphorylation of mannose. That's a big thing. And then it determines it going to make a lysosomes. So now we have our lysosomes. Now the cool thing about these lysosomes is we have to talk a little bit about these proteins, these glycoproteins. These glycoproteins that are all located in these vesicles are very specific. Let's come down and talk about them. So here we have a lysosome. I'm just cutting the lysosome so we can take a look inside of it. All of these are going to be your lysosomal proteins. We also like to call these special types of enzymes. You know, they're really nasty little enzymes. We call them hydrolytic enzymes. They have the ability to cut big macromolecules. And that's really important for their function. So if we were to take some of these actual hydrolytic enzymes, what could they break down? One of them is actually called a nucleases. Oh, that's pretty obvious what this sucker could do then, right? If this is a nuclease, it has the ability to break down what? So this can break down RNA and DNA. That's a really important thing, especially if there's some type of like pathogen, like a bacteria that has RNA and DNA, and I want to break that down, I can do that. Or other things. Another one is proteases. Proteases are a big one. We have the ability to break down proteins. So we can break down proteins. We can use nucleases to break down RNA and DNA. Whew, what else we got in this sucker here? We also have lipases. Lipases. They can break down lipids. They can break down lipids. And we know that multiple different things have lipid membranes. Bacteria can have lipid membranes. Our own cells can have lipid membranes. What else? Another one is called glucosidases. So glucosidases, you obviously can tell that these are going to be breaking down carbohydrates. Glucosidases. These will break down carbohydrates. They will break down carbohydrates. And the last one here is an interesting one. We call these acid phosphatases. We will mention them briefly a little bit later, but I just wanted to write these ones down. These are called acid phosphatases. These are really special to only breaking down bone tissue. So these are actually released by special cells called osteoclasts that contain lysosomes, and these will break down bone tissue, which has a lot of different things within it. But these are the biggest particular enzymes that are contained within the lysosomes. So as you can see, they can break down almost every macromolecule possible, and then even a little extra. But the question is why would they break these things down? In what particular way would they break these things down? We'll talk about that in a second. But another cool thing is that these enzymes, they function at a very particular pH. We know that most proteins and enzymes, at certain temperatures you can denature them, at certain pHs you can denature them. Well, these actually prefer to be at lower pHs. So because these enzymes depend upon a lower pH, there's a special channel or protein pump that's present on the lysosomal membrane. You know what this is? Let's actually zoom in on it right here. So I'm going to zoom in on this particular protein right here. This is called a proton ATPase, which means that it depends upon protons. So let's say here's the inside of the lysosome, and here's the outside of the lysosome. I want protons to be pumped into the actual lysosome, because if it does that, it lowers the pH. I want the pH to be approximately around 5. You want to know why? Because at this pH, it stimulates or keeps these enzymes activated. And that is a really important concept. In order for these enzymes to work, we need their optimal pH to be around this point. So I keep these channels super active and using ATP in order for me to pump these protons into the cell to maintain the optimal pH for these puppies to function. So in order for me to do that, what do I need? I need to utilize a lot of ATP to be able to perform the action of these pumps. That's a really important concept. So we have an idea now that lysosomes are made of glycoproteins, particularly what types of proteins? Hydrolytic enzymes that can break down every macromolecule possible. They're actually made from the transcription of DNA to mRNA to make proteins in the rough ER. They're modified, they're packaged, sent to the Golgi, modified in a particular way, this way, to make lysosomes. They have a phospholipid bilayer that's from the Golgi and the proteins that were synthesized accordingly. Now the question is what would we use all of these enzymes to break down and what type of physiological functions? Let's talk about that. We now know that we have to break down some macromolecules, but in order for us to understand like what kind of macromolecules we're breaking down, we obviously know that it's like proteins and carbohydrates and lipids and nucleic acids, etc. But I want you guys to understand of like what kind of things that we're actually utilizing that for. So there's three particular processes by which cells bring in material, macromolecular material, that needs to be broken down by lysosomes. Because in order for us to be able to break these things down, they have to be brought into the cell, because the lysosomes are found inside of our cell. So the question then comes, okay, how do we get these things into the cell, and what are those things that we're bringing into the cell, and how is that pertinent physiologically? So first example here of how we're going to bring things into the cell is something called receptor-mediated endocytosis. You're like, what the stink is this? This is a really interesting process. So it's kind of said within the name. We're going to bring a particular thing into the cell via a receptor. So here's a receptor that's actually located on the cell. What happens is, let's say, whatever this may be, maybe it's a protein that it binds with this receptor. Maybe it's a bacterium. Who knows? Maybe this is a particular bacterial antigen, right? So maybe there's a particular bacterial antigen that's located on this that binds with this receptor. Or, who knows, maybe it's some type of like lipid molecule. Maybe it's like a lipid molecule of some particular type. But either way, these molecules are binding to these receptors that are located on the cell surface. What happens is proteins called clathrins. You know, there's what's called clathrin proteins. What they do is they come and they bind to the cell membrane and they kind of pull. They use actin and pull the actual cell components here, these receptors and all the things that they're bound to, into the cell. And so what happens is you form like this little vesicle. So what happens is you do this like little endocytosis process and you pull into the cell this vesicle. And here we have on here these receptors, and then on these particular receptors, what do we have bound to it? Like we said, we could have a bunch of different things. It could be proteins, it could be some type of like lipid cholesterol molecule, who knows? Or it could even be like a bacterial antigen of some type. So this is really important. And then once they get into the cell, they then can combine with lysosomes, a primary lysosome, and then they can get broken down. Now, why would that be important, and what kind of cells would this be important? I'll explain to you. This could be important, for example here, I find this very interesting with sometimes when you're trying to do something called LDL receptor uptake. Sometimes whenever you have LDL receptors and you need to down-regulate those receptors, in other words, you don't need as many of those receptors expressed onto the cell membrane, we can bring them into the cell, break them down, or recycle them and then use them for later. So that's one example here. So let's say that, for example, here's this LDL receptor uptake. This could be an example of something called down-regulation. And that's where we would bring these receptors into the cell, which they're bound to whatever it may be, and have them get broken down, and then we can use them later when we need them. That's just one example, An even cooler example here is what's called, you can see these on what's called B cells, so B lymphocytes. So B lymphocytes are referred to as what's called antigen-presenting cells. In other words, let's say that this is a bacterial antigen that binds to a B cell receptor. The B cell receptor will then signal that and bring the bacterial antigens into the cell so that the B cell knows, hey, this is a particular bacteria that I need to alert the immune system of. And so that's another really cool example. But there's an important terminology that comes from this. When we actually take this endocytosis process, when we bring this, whatever it may be, this material into the cell, this macromolecule material into the cell, whether it be through all of these mechanisms, this process that it makes and it pushes these molecules into the cell. This vesicle now is a very important vesicle that we have to discuss. This is now called an endosome. So this is called a endosome. So I want you to remember that, okay? All right, that's one particular mechanism. The second mechanism by which we bring things into the particular cell here is with this next one. So let's say that you have this cell and it wants to eat a particular bacteria, or maybe it's a virus. Maybe it's even a virus of some type. Maybe it's a virus of some particular etiology. And it wants to bring these things into the cell. What it'll do is, it'll actually use actin and kind of make these like things called pseudopods, these little arms. And it'll kind of engulf the bacteria or the virus, the pathogen in general, and bring it into the cell. So then what you get here is, as it forms these things called pseudopods, it sucks this bacteria and virus into the cell. And it makes this vesicle that can contain within it the things that it tried to eat. Look at this. Look at this. Isn't that cool? What is this process called, and in what kind of cells would this be critical and important to know? This is something that you want to know is called phagocytosis. And I'd say that this is probably one of the biggest indications and reasons for having lysosomes, honestly. So this is a really, really big one. And this is pertinent in what's called white blood cells. So this is really, really important for particular types of white blood cells. What kinds of white blood cells? Things that you should know is things like macrophages. So things like macrophages. Another one is called neutrophils. Neutrophils are another one. Another one is called dendritic cells. These are really, really common types of cells that love to eat certain types of pathogenic material and bring it into the cell. And obviously you can understand that in this example I bring in proteins and lipids and carbohydrates, and this one I'm bringing in maybe a bacteria or a virus that has nucleic acids. Their membranes could have lipids in them. They could have proteins that are involved in all of these structures. And guess what? I got the enzymes in my lysosomes that can break all of them down. So you see why this is a really cool concept? All right. The last one here is a really interesting one, and this is called autophagy. This last one is called autophagy. And it's another way that we don't necessarily bring things into our cells. We just actually membranize. We form a membrane around already present organelles and proteins that are defective and old inside of our cell that's already there. So in other words, let's say here you have an old mitochondria. All of these things are old, they're defective, they're just not working as well anymore, okay? And so what happens is you actually start to form this membrane around these old defective organelles and proteins. So what I'm gonna do is I'm gonna start forming kind of a membrane, this makeshift membrane here. And then what happens is, after you form the makeshift membrane, it'll eventually completely membranize, I hope that's a word, membranize, and enclose all of these particular organelles. When you do that, that's really, really important because you're kind of internalizing and forming a membrane around these defective organelles that you want only those ones to get destroyed, broken down. We don't need them anymore. Guess what? Lysosomes can break down proteins. Lysosomes can break down lipids. Lysosomes can break down maybe nucleic acid molecules. So it can break all of these things down. An important terminology for both of these. So this one here, where we actually, so this one we call an endosome. This one where we bring these particles in by eating it. It's called a phagosome. It's called a phagosome. And this last one here, where we actually internalize old defective organelles and proteins, this is called a autophagosome. Phagosome. This is really, really important. Now, once we've done all of these particular processes, we've now taken a way of either bringing things into the cell and enclosing them in a particular vesicle that can fuse with the lysosome, or things that are already old, defective in the cell, we formed a membrane around them, and now we can take these to the lysosomes. So now this is the next step, my friends. So now what we're gonna do is we're gonna take this phagosome, this endosome, and this autophagosome, and we're going to now fuse it with lysosomes. So let's come down for a second. Here we have the first one. Here is our endosome. What we're gonna do is we're gonna take this endosome and we're gonna fuse it with a lysosome. So each one of these this is going to occur here. So we're going to take these and we're going to fuse them together. So you see how this is what's called your primary, I'm going to write all of these down here. This is your primary lysosome, primary lysosome, primary lysosome, okay? Each one of these, the endosome, the phagosome, and the autophagosome are going to fuse with this primary lysosome. Once it fuses with the primary lysosome, guess what? All of the enzymes in here, the nucleases, the proteases, the lipases, the glucosidases, all of these are going to break down. They're going to rip that stuff apart. And they're going to destroy all the proteins, the lipids, the nucleic acids that are associated with the bacteria, the viruses, maybe some of the lipids and proteins that are present from that we brought into the cell, maybe all of those things that are associated with organelles. All of these things are going to get broken down. And as a result, at the end of this, after you've broken down all of these things, this is what it looks like. You've now broken down all of this macromolecular material into maybe a smaller monomers or kind of small residual particles. You can do two things with this, but we have to know a term now. This term here is that this lysosome is called, we're going to point to this one, this is called your secondary lysosome. So your secondary lysosome is the lysosome that's already fused with each one of these, the endosome, the phagosome, the autophagosome, and it's broken down all that material that's present in them. Now, a couple different terminologies here before we go to the next step here. Whenever you take an endosome, a phagosome, an autophagosome, and fuse them with a primary lysosome, and right when the moment of them fusing, so this point here, these are called specific types of things that you have to understand. The secondary lysosome is all the broken down products after these things have fused. But this point right here that I'm going to highlight, so this is going to be 1, this is going to be 2, this is going to be 3. These have specific names. This one here where the endosome and the lysosome fused is called, believe it or not, it's not too crazy, endolysosome. Okay? This one here, where the lysosome and the phagosome fuse, this is called a phagolysosome. And this last one here, where the lysosome and the autophagosome fuse, is called a autophagolysosome. Not too crazy and out of this world to understand those particular processes. Okay, so with all of these, we fused each one of these things, formed these intermediates. After we formed these intermediates, they broke down all the material, all the macromolecular material, and formed a secondary lysosome. The question then comes, what can we do with all this residual material? Well, I could have taken proteins and broken them down into amino acids. I could take lipids and break them down into fatty acids. I can take nucleic acids and break them down into nucleotides. I can take carbohydrates and break them down into monomers. So what I could do is, I could push and export all of these different molecules, such as small carbohydrates, such as small amino acids, such as small lipids, and etc. You get the point. All of these can be pushed out into the cell and they can be used in metabolic reactions. So two things can happen with these. They can go and they can be used in metabolic reactions, or they can kind of just sometimes, believe it or not, sit there and they can make these kind of like little old inclusions. And sometimes we actually give this a very specific name. We call it lipofuscin. And you see this. This is usually representative. The more you have of this lipofuscin bodies in the cells, it's more indicative of age, increasing age. Obviously that would make sense. As you break more things down, you're going to have more lipofuscin. And so you can see this very commonly in three particular organs. One is you can have a lot of this lipofuscin in the heart or in the cardiac tissue. You can have a lot of this in the nerve tissue. And you can have a lot of this in the hepatic tissue. Okay, in the hepatic tissue. But this is something that you could potentially see. So if we push it out into the cell, we can use it for metabolic reactions or store it as this like lipofuscin kind of material, which is usually associated with increasing age. Let's write that down. Associated with increasing age, that's very prominent in these particular tissues. The other thing that we could do is we could take this secondary lysosome and say, I don't need any of these things. I'm going to go ahead and fuse this with the cell membrane, and then I'm going to release some of these materials out of the cell. So then what I can do is I can literally release, I'm just going to draw a couple of these dots here. I'm going to release some of these molecules into the actual external environment. This is sometimes kind of important. When we get into immunology, we'll talk about this later. Neutrophils love to do this. Neutrophils will take a bacteria, eat them up, and then spit out some of their antigens and spit it into the lymphatic system so that it can go to other immune system cells and they can pick it up. Sometimes your lysosomes will release special enzymes, believe it or not, right out into the extracellular fluid. Remember I told you that in osteoclasts, bone tissue, guess what they can release right onto the extracellular space and break down bone tissue? They can release acid phosphatases. So you get the point here that what I'm trying to get is that we can take all of these ways of bringing in macromolecular material into the cell. Once it's brought into the cell, they fuse with lysosomes, make these intermediates. They get broken down accordingly to each particular enzyme in the macromolecule. The residual lysosome is called a secondary lysosome. It can spit things out, or it can spit it out into the cell or spit it out of the cell. That's the big things to understand. The last particular function that we have to talk about lysosomes is called autolysis. All right, my friends, last part is autolysis. So basically this is where the cell eats itself. It's essentially what it is. So autolysis is where the cell is literally eating itself. So what happens is usually you have such extensive cellular damage. So let's say that this cell has gone under extensive cellular damage, like beyond repair, extensive cellular damage beyond repair. When that happens, your lysosomes receive a pretty intense signal. So let's imagine that you have extensive damage to the DNA, you have extensive damage to particular organelles, you have extensive damage to particular proteins, and what happens is, after this extensive damage occurs, it then stimulates and activates these lysosomes. It sends them powerful signals. And you know what the lysosomes do? They're like, oh shoot, this cell is beyond repair. We're gonna have to finish this up. And then what it does is it literally releases its contents, all these nasty hydrolytic enzymes, directly into the cytoplasm. Now imagine having these hydrolytic enzymes that can literally break down every single macromolecule just uncontrollably released into the cellular environment. I have proteases, they can break down every protein. I have lipases, they can break down the cell membrane and the membranes of organelles. I have nucleases that can break down DNA, RNA. I have glucosidases that can break down all the different types of carbohydrates. This cell will literally start to eat itself. And that is the process here, where this will undergo an intense process where the cell will literally cause cell to eat itself. Eats itself. I mean, this is literally, in layman's terms, what's literally happening. And it's a pretty interesting process. So it is a little bit different from something like apoptosis, but again, this is when there is such extensive cellular damage beyond repair that the lysosomes receive signals to release their hydrolytic enzymes into the actual cytoplasm and start breaking down all the different types of organelles. This is autolysis, and that covers the structure and function of lysosomes. I hope it made sense. I hope that you guys enjoyed it, and as always, until next time.